Alrighty, we're back at it. Now we're going to talk about the more specific adaptations to the immune system. This is known as the adaptive or specific defenses. These are the kind of defenses that you're more used to, uh, the kind that you get when you get vaccinations. You create a specific uh, group of cells and antibodies against a very specific type of disease or pathogen with specific defenses. As we've been through in the first uh, part of this lecture, we went through uh, what are known as the innate defenses. Innate defenses are non-specific. They don't care what kind of foreign invader comes in the body. They're going to attack it, they're going to identify it as foreign, and they're going to deal with it in that sense. This is the more specific kind where it's a very specific, and in fact, so specific that we're dealing with specific kinds of molecules on the outsides of the virus, of the bacteria, and these can vary from what is known as a strain of virus or a strain of bacteria. So you can have essentially, these days, you have a coronavirus going around. Well, it's a specific kind of coronavirus that has not been seen before. That's why it's called a novel coronavirus. But there are lots of other kinds of coronaviruses that you have seen before. They're called colds. There are hundreds and hundreds of different varieties of these, and that's because they have different molecules on the outside that are presented on the outside of the virus, and those molecules are referred to as antigens. If your body has seen the antigen before that's presented on that particular virus, you have had an immune response to that virus and you have developed a secondary immunity to that virus, an adaptive immunity. If you've never seen it before, if nobody's immune system has ever seen it before, then you have not created a secondary immune response, so you're having a primary immune response. And it takes much, much longer to ramp up the body's defenses to mount a significant immune response. That's why this particular virus that's going around now is so uh, infective and contagious, is that nobody's bodies has ever seen this, and that's why it's referred to as a novel virus. So the antigen is the molecule on the outside of the bacteria, the virus, the pathogen, that are recognized by your immune system as what is known as non-self. That means it's not you, because your immune system floats around and has to live inside your body, and it has to be able to identify who you are versus who you aren't and respond appropriately. And it's specifically designed to do that. So these adaptive responses are carried out by lymphocytes and macrophages, and there are two specific kinds of adaptive defenses. You can have a cellular immune response that's performed by immune cells, and then there is what's known as a humoral immune response performed by antibodies. And these two kinds of adaptive defenses are performed by different types of cells in the immune system, and we'll talk about those next. So here's the pathway that a pathogen gets interrogated by your body. The first thing, of course, is that we try to keep it out, right? Mechanical barriers, skin, mucous membranes, we already talked about those. You're trying to keep it out. If you can't keep it out, so that's the first line of defense. So the second line of defense is that the pathogen gets in and you have a non-specific response to that. And that is, as we talked about before, the chemical barriers, natural killer cells, inflammation, phagocytosis, fever. All of those things are non-specific. That means almost any kind of non-self entity that your body sees coming into you as a pathogen, your body's going to react to it. There's a very standard set of reactions. If that is not enough, or even if it is enough, you still would like to react faster to that in the future. And so then you get into this third line of defense. and that's the cellular immune response and the humoral immune response. So we'll describe those now. So antigens, again, let's talk about what is an antigen. An antigen, you have to think the immune cells of your body, they're floating around, and a virus or a bacteria invades, and it starts to float around too. You know, it's not like your immune system can look at this virus or bacteria and say, aha, I know what you are. It's like a blind person, or if you walked into a very, very dark room and you couldn't see anything, what would you do? You'd have to hold your hands out, you'd stick your feet out, you'd try to feel around, right? You'd feel for familiar objects. You'd say, oh yes, that's my dresser. Oh, there's my bed. And then you'd say, oh, what's this thing over here? If you didn't recognize it, you might hang on to it. You might grab onto it. You might pull away from it, depending on the situation. Well, your immune cells do the same thing, but they have to figure out this protein, this part of that protein. Yep, I know that protein. I know that polysaccharide, glycoprotein, glycolipids. All those things, they identify those as self. They say, fine, that's good, that's good, that's fine. No problem there. But then it comes across an antigen that it doesn't recognize, and it says, whoa, 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 what's this? And it goes after it. It attacks it. It defines it as non-self. The tiny little bits and pieces of an antigen are referred to as haptens. It's not so important that you really understand what a hapten is. A hapten is a small molecule that by themselves don't raise an antigenic response, but if that hapten is attached to the outside of a virus, the outside of a bacteria that your immune system recognizes as foreign, then that hapten can stimulate or spur on an immune response. All right, so these cells, right, they're lymphocytes. Lymphocytes are the cells of the specific adaptive immune response. And there's two kinds of lymphocytes. And remember again, never let monkeys eat bananas. Never is neutrophils, those become macrophages. They're one kind of phagocytic type of cell. Next in line in terms of the number of cells that you produce are lymphocytes. And lymphocytes come in two flavors here, the T lymphocytes and B lymphocytes. They're produced throughout life. They start in fetal development. Red bone marrow produces these cells. They're produced by stem cells of these. There's a slide coming up where we're going to talk about the stem cells, where they come from. Some of these stem cells settle in the thymus. The thymus specializes those particular lymphocytes into what are called T lymphocytes, or T cells, thymus-derived lymphocytes. They make up, as you see there, 70 to 80% of circulating lymphocytes. So, 3/4 or so of the lymphocytes that are floating around your body are T cells. There are various kinds of T cells. They split up into different kinds, different types of T cells, and we'll talk about what those kinds are. Some of these T cells end up in lymphatic organs, lymph nodes, thoracic duct, the white pulp of the spleen. Remember, the white pulp is where white blood cells accumulate, and the red pulp is where red blood cells accumulate in the spleen. And then the other kind of B lymphocytes. B lymphocytes, they're named for, the B is a bursa of Fabricius in chickens. That's where they're first identified. Doesn't really matter what that is. That's just the B. The B stands for the bursa of the Fabricius in chickens. T cells, much more easy to remember. T cells are matured in the thymus. So the B cells, after they release from the bone marrow, they make up the other 1/4 or so of the lymphocytes. Of course, they're abundant in lymph nodes, just like T lymphocytes are. Spleen, just like T lymphocytes. And bone marrow and intestinal lining, which is a little bit different than T lymphocytes. So here's a chart. You've seen this before. This chart comes from hematopoiesis. Remember way back when we were talking about blood clotting, and we were focused on the left side of this slide. The left side describes where do erythrocytes come from, and where do platelets, thrombocytes come from. That's what we were talking about before. Now we're talking about the right side of the slide. Right side of the slide is you still have the hematopoietic stem cell that creates all of these blood cells. Well, the white blood cells come from the right side of this. And in particular, the T and B lymphocytes that we're looking at now, they come from the lymphoid stem cell. And then there's a maturation process. Remember, the T lymphocytes will have to mature in the thymus. Thymus is a very large gland. When you're a baby, when you're very young, it's a large gland. You're developing a lot of T lymphocytes. As you age, it reduces in size. We say it atrophies. So the thymus gets smaller and smaller and smaller as you mature fewer and fewer T lymphocytes as you age. And then you can see the pathway of the B lymphocytes as well. So here's the origin. They're all made in red bone marrow. Right? Red bone marrow found in flat bones in the proximal ends of long bones, like that bone there on the left. Looks like the head of the femur. That's where red bone marrow is. So you're making blood cells in those areas of the body. Those lymphocyte precursors are released into the blood. Some of them are transported to the thymus. They mature in the thymus, and they become T cells. Some mature further on in the bone marrow. They become B cells. B and T cells both end up in lymph nodes, and we went over that in lab. And if you need to review that in lab, your textbook has a great picture and description of this, but your lab book in lab exercise number 49 is really good at describing and having pictures of what a lymph node is and what it looks like. But that's where these cells are found, and they will help to spur on the more specific immune response to any infection that you have. All right, so here's our tables. You know all about our tables now. So the origin of the undifferentiated cell, they both come from the red bone marrow. No surprise there. The site of differentiation is different. T cells in the thymus, B cells in, so you can think of T as in thymus, thymus, B cells in bone marrow. That's where they mature. Primary locations that they're found, they're all found in lymphatic tissues, of course. 75%, 70 to 80% of the circulating lymphocytes in the blood are T cells. 20 to 30% are, 1/4 is about the circulating lymphocytes in blood. Those are B cells. So the primary functions, they're slightly different here. T cells provide, as they say there, it's cellular immune response. T cells interact directly with antigens or antigen-bearing agents, and their mission in life, T cells' mission, is to destroy either the cell that has the antigen or the infectious agent in it, or to destroy the agent itself directly. B cells, their focus is to produce antibodies. They are antibody machines, antibody manufacturing warehouses. They produce tons and tons and tons of antibodies. And we'll talk more about their production of those antibodies in a moment. But that's what they do. They produce antibodies. We're going to talk about what antibodies do. So B cells produce antibodies. T cells kill the infected cell, or they kill the pathogen itself directly. So a lymphocyte has to be activated. What does that mean? It has to be activated. Well, what we have with T cells, T cell activation requires that antigenic fragments be attached to the antigen presenting cell. Holy cow, what the heck does that mean? I'm going to show you a video here. Let's go to this video and it'll make more sense. So let's take a look at this.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells.
So did you see what that cell did, right? It was just a macrophage. It was part of the primary or secondary immune response. It saw a foreign invader, didn't care what it was, it ate it up. It's a macrophage, that's what it does. And then it ripped it up, it used its lysosomes with enzymes inside the lysosomes, tore up the antigen, didn't care what it was, breaks it up into little pieces, and then those little pieces it puts out, displays on the outside of the cell membrane. And what it's doing is it's sending off a neon light. It's saying, I found something. I killed something. Here it is. Here's a piece of it. So, back in the old days, when the way old days, when you invaded the country or town of your enemy, you you would take the the heads, the lords or the chieftains or the heads of that particular town or village or whatever. This is disgusting, of course, but they'd cut their heads off and they'd stick them on a spike, and they would put this big, tall, 6, 7, 8 foot long spike of wood into the ground with the heads of their enemies on it. And it was a display to everyone else. Look what we did. We killed everybody in this village. We took it over. Look what we did to them. You should fear us. In a similar sense, macrophages kind of do that. They tear apart the virus, they tear apart the bacteria, they cut it up into little pieces, and they take the heads of those viruses or bacteria and stick them on the outside of the cell. But what they're doing is they're presenting it. They're taking a piece of that antigen and they're presenting it to the rest of the immune system and saying, look what I found. Come and help me out. All right, so now from here we go. So that's called an antigen presenting cell.
An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from
So the cytotoxic T cell is another subdivision of the T cells that are uh, around, that are floating, circulating. Cytotoxic T cell is what you would think it is. It is cytotoxic. It is toxic to other cells. It's going to kill other cells. That's its mission. Ultimately, that's what T cells do. T cells, as I mentioned before, they directly kill either infected cells or they kill the virus or bacteria that are present. The B cell, on the other hand, the B cell has a different role. The B cell is going to make antibodies. Antibodies are red flags. They're fluorescent markings onto the virus or bacteria that has infected the the, the bloodstream. What that means is uh, that B cell will make antibodies. That antibody is specific for the antigen that has been presented to it. It will make lots and lots and lots of antibodies, which will attach to the foreign invaders. They'll attach to the virus, they'll attach to the bacteria. They are marking it for destruction. They are telling the rest of the immune system, kill this thing. That's what an antigen, I'm sorry, that's what an antibody does. An antibody marks the foreign invader as a foreign invader. And it says, kill this thing. That's where a cytotoxic T cell will come along and say, I recognize you as the foreign invader. I'm going to kill you. Now, the killing can happen either cellularly The cell itself can kill the organism, or it can, what we refer to as mediate the destruction of that organism through chemical destruction. So we'll continue on with the video now.
From this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell.
So essentially, this cytotoxic T cell recognizes the cell that has been invaded by a virus or a bacteria. That cell, in due time, chops up as many viruses or as many bacteria as it can, and it presents the antigen on its surface. But this is a normal body cell. This normal body cell is saying, it is literally saying, kill me, destroy me. I have been invaded. I cannot be saved. You must destroy me. The cytotoxic T cell comes along and says, it feels for its specific antigen binding site. It feels that antigen on the outside of that cell and says, aha, I see your message. I will now kill you, which sounds a little dramatic, but that's how it works, right? And so this cell, this normal cell up here, has been invaded, and it's telling the cytotoxic T cell here, kill me. Cytotoxic T cell does just that. So this cell will die. But of course, now it can no longer produce any more viruses or any more bacteria, which is really the virus or bacteria's point in taking over that cell. So this cell will no longer be able to make any more viruses or bacteria. Then that's what you want. That's what you want to happen.
Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection.
Okay, so you see where B cells themselves have an antibody on the outside of their cell. We're going to talk about that in a minute, about where that comes from and how that works. But they have this antibody on the outside. It recognizes the foreign pathogen, and then some of those B cells transform into what are called plasma cells. It's the plasma cell that is the actual antibody manufacturing machine, antibody manufacturing factory. And these plasma cells will put out as many as 2,000 antibodies per second. 2,000 per second. Every one of those plasma cells can put out that same antibody, and that antibody is specific for the specific antigen that it's looking for. So these homing signals are spread out through your body, through your bloodstream. They're looking for a particular antigen, and they're going to mark it. They're going to mark it for destruction.
Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So that's precisely the reason that you get vaccines. Vaccines present small pieces of a virus or a bacteria, the antigen, the antigenic region. They present it to your immune system, but they don't present the whole virus or the whole bacteria. That would make you sick. So we present this little antigen to your immune system. We say, hey, look at this little piece. Your immune system says, I don't like that thing. It marks it with an antibody produced by a plasma cell, and then the remaining B cells that remain behind create a memory of that antigen. And so now, instead of having one memory cell or one B cell that is specific for a particular antigen, now you have hundreds of thousands or millions of memory B cells. And now the next time you see that antigen, now you're going to react much faster. Rather than having a tiny little reaction and having to build up your immunity, you already have it. It's raring to go. When you see that thing the next time, so coronavirus comes around the first time, it's a novel coronavirus. We're all naive to it. We've never seen it. Our immune systems don't know what to do. But they create a response. You create memory B cells, and the next time this thing comes around, you're already wise to it. Your immune system is ready to take it on. And that's why the secondary response is much faster after you have been exposed to a particular antigen.